A very good afternoon, everyone. I am A. Revati, working as an assistant professor in Sir CRD College of Pharmaceutical Sciences. A very warm welcome to each one of you to the fifth session of our faculty development program. We are gathered here today to start our journey of growth and learning together. Today's program has been meticulously designed on research excellence, tools to compete and achieve, to provide you with the necessary tools and knowledge to excel in your roles as an educator. We understand that the challenges you face and the importance of continuous improvement in the rapidly evolving educational landscape. During this program, we encourage you to actively participate, engage with your peers, and take advantage of the valuable resources and expertise available. We believe that the knowledge and insights gained here will not only benefit you individually but also have a positive impact on our institution and most importantly on the students we serve. Let's make the most of this opportunity to collaborate, share experiences, and explore innovative teaching methodologies. Remember, the pursuit of knowledge is a lifelong journey, and we are here to support each and our other in every step of the way. Now, let's begin our session with an introduction of our today's guest speaker, Dr. M. Murli Krishna Kumar sir, and he is working as an associate professor in pharmaceutical chemistry department at AU College of Pharmaceutical Sciences. Now, I would like to invite K. Shravani ma'am to give the introduction of our guest speaker. Madam.
Yes. A pleasant good afternoon to everyone. This is Ramli Koralla, Assistant Professor in Pharmaceutical Chemistry from Sir Reddy College of Pharmaceutical Sciences. I am pleased to introduce Dr. Mutyala Murli Krishna Kumar, a distinguished academic and researcher in the field of pharmaceutical chemistry. Dr. Mutyala Murli Krishna Kumar has an impressive educational background, extensive teaching and research experience. He holds a PhD in pharmaceutical chemistry from Andhra University, Visakhapatnam, and an M.Pharm in pharmaceutical chemistry also from Andhra University, where he received high academic honors. His academic journey has been marked by his commitment to teaching and research. He has held the position of assistant professor at the College of Pharmaceutical Sciences, Andhra University, Visakhapatnam, for several years, where he has made significant contributions to the field. His expertise in medicinal chemistry, advanced instrumental method of analysis, and pharmaceutical research is evident in his dedicated work. In addition to his teaching responsibilities, Dr. Murli Krishna Kumar has been actively involved in research projects and has received grants for several significant projects. Notably, he has worked on projects related to biofuels from marine microalgae, the synthesis and screening of novel anti-tubercular compounds, and the development of pharmaceutical agents against Mycobacterium tuberculosis. Dr. Murli Krishna Kumar sir, research contributions extend to patent applications with an Indian patent title, Design, Synthesis and Pharmacological Evaluation of Benchimidazole and benchimidazolium derivatives to his name. He has also published 19 national and 26 international journals on topics ranging from anti-tubercular drug discovery to bioactive natural products. In the academic research and community, he is well respected and recognized for his work. He has developed open source software AU Docker to facilitate virtual screening and ligand optimization, making his research accessible to a global audience. His impressive career has also seen him to take on administrative role as the director of the NMR Research Center at Andhra University and as a placement officer at AU College of Pharmaceutical Sciences. He is not only a dedicated educator and researcher, but also an active participant in various workshops, seminars, and conferences. His contributions to the field of pharmaceutical chemistry, especially in the context of tuberculosis research, have been significant. We are fortunate to have you with us today, and I am confident that his wealth of knowledge and experience will greatly benefit our event. Please join me in welcoming Dr. Mutyala Murli Krishna Kumar to this session. Thank you for letting me to speak about an eminent personality of Andhra University.
Thank you, madam.
Thank you, sir.
Now I would
Thank
like
you,
to hand over the
Shravani, for introducing me. Lavanya, can I start the session?
Yes, sir. Yes, sir. You can start.
How can I
Thank you,
share?
Professor.
See, how to share this one? I don't know.
Sir, at the top of the screen, you have share button. Beside the camera and mic button, you are
Yeah,
having
share
a
is
share.
there. I'm sharing the entire screen. I think it would
Yes,
be
sir. On the screen, you have to open
better.
your PPT.
So, can you see my slides?
Yes, sir.
So, very good afternoon to all of you. So, myself, Dr. Murali Krishna. Can all of you hear my voice? Am I audible?
Yes, sir.
Okay. So, thank you, Nagaraj Garu, for giving me an opportunity to interact with young faculty virtually. And also congratulate you for choosing right topic. Research excellence, tools to compete and achieve is my topic today. So where I just wish to let you know the current scenario of research work, where exactly things are going right, where exactly things are going wrong. And as faculty, as the future of pharmacy, how you all can contribute in enhancing excellence or achieving excellence in your chosen fields. First of all, faculty position itself is a little confused. Okay, so regarding as far as career is concerned, as far as other benefits are concerned, one will be in a perplexed situation all the time. Faculty is a teacher or research or admin position. Where can we go? We always will have, because we are all professionals. Okay, we are not doing anything like a service. We are all professionals. We have chosen a field where we distribute knowledge, we spread knowledge to the younger generations. But it's a career. Okay, at the end of the month, we have to earn salary, and we need to see that as we are aging, as we are progressing, we need to see that our career is strengthened, our career is growing. So career growth and career development, these are the two terms where we need to have absolute clarity. Career growth is like, for example, you are an assistant professor, you wanted to become the dean of an institute or a principal of an institute or a director of a research organization. So that is a vertical growth. In case of career development, so as an assistant professor, how can I grow? So here you need to see how you can adopt newer technologies, how you can engage yourself in newer activities and newer outcomes you can expect being in the same position. So these are the two situations completely different where one need to be very, very clear. I could see people opting for specific positions in order to see that their career will grow vertically but not horizontally. Okay, like an assistant professor, he can aim to become a principal, absolutely no problem with that. But if as a faculty and in other fields, if faculty is a failure, he cannot lead the institute. So as an assistant professor, he is a must to scale the career. Means career development is the usually it forms the foundation for your career growth. So this is where one need to be very, very clear, and how research or experience in admin along with your teaching capabilities, how these helps you achieve these two objectives of career development, career growth. So here again, another interesting question, that is, faculty need to do research work. And immediately you will ask me, why do we need research? See, you're a faculty or a teacher. Our job is to see that young brains of students are filled with knowledge. We offer them clarity on subject. We clarify their doubts and we'll show them a direction where they can excel. Okay. So here, is there any need for research, or why are we supposed to do research work? In most cases, when we talk to each other, when we talk to colleagues, when we talk to students, why you are doing research work? Sometimes they say like, okay, I have to get a PhD. I'm doing my master's thesis or bachelor's thesis, or I'm working for a paper. That is not research work. Okay. So if our objective is limited to only getting a degree, only getting a student's thesis work, a faculty is not actually doing research work. He is actually mass producing paper works. We usually have to link ourselves to do research work with these objectives, to provide solutions to complex problems, to investigate laws of nature, to make new discoveries, to develop new products, to save costs, to improve our life, and to satisfy the human desires, some of them. Okay. So if you have higher objective, then you may claim that you are doing research work. Otherwise, one is actually doing paper work. Okay. So this is where one need to be very, very clear. Am I doing paper work or research work? One cannot merge these two things together. A research work can result in publications of good quality, but papers won't reflect in the kind of research work one is doing. Okay, so this is an interesting situation. You will understand further. So people used to have this kind of a confrontation, like how to do research work. So, sir, I am doing papers, I am publishing papers, I am doing so much of work, and you are saying that they are paper works, but not research works. See, excellence comes out of proper planning, proper objectives, proper execution, etc. So in case of research work, we have a sequence of events, like first we go through the existing knowledge, literature. So we commonly use the word literature. That means existing knowledge. Let's say when you are pursuing some work, going through a research article, going through certain situation, talking to a learned person, all of a sudden you get an idea. Okay, so origin of idea is the existing knowledge and interpretation of the existing knowledge by your brain. So in a succinct manner, to present everything in a nutshell, we call that one as literature search. Literature search does not mean just googling, using Google or using a research paper and reading a book. It is also interacting with people and nature and environment around you. And once you got that idea, you conceive the problem. Okay, so you will see like, okay, so something is untouched, so there is some issue which I can address, or something will strike on your mind, saying that you have a peculiar point in that problem which gives you immense interest, and that gives you immense interest, immense focus, and immense encouragement. So that is what we call it as a problem to be pursued. Okay, so not just like you are using starch as a binder, so I will use cellulose acetate as a binder. That people won't call it as a research work. Okay, And then comes planning and design of experiment. Now you have a problem with you, with the existing knowledge. You are an expert in a specific field. By using your analytical capability, existing knowledge and experience, you will design an experiment. You will plan a research work. Okay, so that is the stepping stone to do research. So once your plan is over, then you have to execute it. And then the execution gives you information in terms of numbers, in terms of words, in terms of statements. So whatever the output may be, after execution of the experiment, you will have to sit down and do the data analysis. So once the analysis is done, it will let you know clearly whether your thought process is going on properly or not, and the experimental conditions, whatever you perceived, are actually working or not. Then you will do revision and renewal. Let us say if any course correction is needed, that you will probably do here. And then the outcome that you got, okay, is actually translated into a publication, a patent, or a product. So this is the sequence in which usually research is done. So now let us see, this is, you can say like, sir, this is what exactly is done by any researcher. Now let us see where exactly excellence comes. Okay, every one of us is a researcher, but where exactly excellence comes? Excellence in literature collection. So how exactly we do the research, literature search? So normally what happens in our regular labs is like we give a topic, student will go, or researcher will go, or we ourselves will go, sit before the computer with internet, and then simply click the words in Google and then search. In the top 10 hits, whatever are obtained, we just take them as literature. Okay, so that is not what exactly is literature search. There are so many other ways where you have to refine yourself. I don't mean to say Google is bad. No, it is not like that. But the way how you are using Google is exactly you need to know. First one is define your research question. So what exactly you are looking for? You have to be really good at framing the question, framing the phrase, framing the question, framing the words, framing the combination of words. Okay, so this is a very important trait every researcher need to develop. Let's say you are looking for cardiotoxicity of a specific drug. So how do you frame your question? Because regarding computer, a rule.
Sir, your voice is not audible, sir. Sir, Murli sir.
Hello, madam. Backside some resound is coming, madam. Please, can you?
Hello, can you hear me?
Can you manage
Sir,
that one?
now it is. Sir, now it is audible, sir.
Okay, thank you.
Please manage that one. Backside sound is coming.
Okay,
so
sir.
Here, can you hear me, man?
Yes, sir.
Hello?
Yes, sir. Continue,
Okay.
sir.
Yes, sir.
So
Yes,
in
sir.
the literature collection, as I told you, the keywords that we are giving are very, very important. So you have to be so specific as far as the definition of the problem is concerned. So here you have to define your research question properly. What are the words that you have to keep? What are the words that will give ambiguity? You have to eliminate. Then relevant search terms are needed. Okay. And then develop your research plan, because let us say you are searching and the answers are ambiguous and answers are not giving you properly. Then you have to see whether you are using right platform or not. If you have a problem with natural products as your research field, then if you are using Google is an open platform. Okay, so it may give you a holistic view of the research output that what you are looking for. So the search that you have to do is in a different database. So where exactly you have to go and then see that you have to develop. Let's say you are working in Sudix field, then those scientific publications like American Chemical Society or any other SciFinder or any other database that you can choose. It all depends upon your research question. And then test and refine your search, because searching in Google is not troublesome. It is straightforward. It is very quick. So in the beginning, when you are trying, let's say your words are not matching or the computer is interpreting it in a different way. Now you have to refine your search terms. So here, select relevant databases, because as I told you, Google is an open database. Okay. So let us say you wanted information on chemicals. Then the database that you are supposed to look into is actually SciFinder Scholar, where the comprehensive information is available exclusively on chemicals. And then once the literature search is over, you are supposed to summarize your literature search in a document. Normally, let's say you are working on a specific problem and you are searching in a database, appropriate database, you have some hits, like you are searching for cardiotonic activity of natural products. That is the search term that you have given. And the database is giving you 15 different research articles which fits into your search. Then you have to see that all the 15 articles are downloaded, and in a notebook or by using EndNote or any other advanced technological apps, you can summarize the data, so that in a chronological order or in an alphabetical order, you have to summarize whatever literature search that you have done. And also you have to consolidate the output that you have obtained. Okay. So cardiotonic activity of natural products. So how many plants are known to have cardiotonic activity? Because we have studied cardiac glycosides. Apart from that, we don't have any other cardiotonic drugs available in our textbooks. Maybe so many hundreds of molecules may be there in the literature. So that gives us a holistic idea on distribution and existence of cardiotonic materials. So that entire thing is to be summarized at the end. And also we need to keep the literature in an appropriate manner so that we can go back and forth whenever it is needed. Second one, whenever we use the word excellence, okay, in case of literature search, one need to be very, very sure as far as existence or non-existence of a information. That means, let's say you are working on a plant. Okay, so you are working on Datura plant of a new species. You think like the plant is completely new as far as species is concerned, and now you have started. Okay, and what happens to your literature search? You have initiated your research. You also have to parallelly monitor the current literature, because nobody knows when and where this plant is available. There is every chance that someone else may be working on this plant. Okay, you have to closely monitor the literature, so that whenever something is happening somewhere on similar kind of a plant, you can be very, very careful. For example, you have isolated a compound, someone else has also isolated a compound and published it. Then what will happen to your research work? So that's the reason, as far as literature is concerned, you have to continuously monitor the literature. It is not like literature search is a one-day job or 1 month job. No, it is not like that. Throughout the research work, you have to continuously monitor what exactly is happening throughout the world as far as your specific problem is concerned. Next one is, once the literature is done, something strike on your brain. Now you have to conceive the problem. So what exactly is the problem that I can pursue? Okay, So already certain drugs are there which are having cardiotonic activity. Let us say cardiotonic activity is your topic of interest. So what actually is considered as a problem? Let's say digoxin. You are a Sutix person and digoxin is the topic of choice for you. What is the problem you are trying to answer? Digoxin has a stability issue. So stability issue is there. So I wanted to develop a sublingual tablet or a patch form so that instead of giving intrathoracic or kind of injections, we can simply give a oral solution or something like that. So normally, how do we do that? How many types of problems can be perceived? You can have an alternative, a number of alternatives. Problem is there which is visible that needs to be solved. And somebody is frustrated in using a tool, that is a problem. Something is there which needs to be changed. For example, we are using sulfuric acid as a reagent. We wanted somewhat grainer solution for that. We are using cadmium. Maybe that is not grainer. So we wanted to use some other catalyst. Let us say somebody is using cyclodextrin for formulation. So we need some other solution for that. Something needs to be changed. So disagreement over the action. So disagreement over the action means in the literature, somebody is saying that rabbit has only one leg. Now you are very sure that rabbit has 2 legs and 2 hind legs, 2 front, 4 legs are there. But here, there, they are mentioning only one leg. There is a disagreement in the principle. You can pursue that and prove that the existing data is wrong. A fault has occurred somewhere that can be repaired. Difficult to reach goal means somebody is attempting to obtain 80% yield in a synthesis. 80% yield in synthesis. So they are ending up in having only 40%. So you can add something to that. You can do something with your thought process and bring development in that process so that you can achieve 80% yield. Unexpected outcomes. Unexpected outcomes are also problems. Okay. So like sildenafil citrate, unexpected outcome is a useful one actually. So that is pursued. And in case of diphenhydramine, it is supposed to be antihistaminic activity, but it is actually, it causes drowsiness. That is an unexpected outcome. It is a problem. Somebody pursued it. So we have fluoxetine, an antidepressant drug. Very interesting. A barrier to progress. A barrier to progress means there are certain experimental conditions which are actually curtailing our freedom to execute certain things. So they are to be eliminated. Okay. So this is what exactly are the kind of problems that we can pursue, we can bring out from the literature search, in the literature search. So wherever your psychology fits, wherever your environment fits, wherever your heart goes, all that particular topic you can choose as your research topic. Hello, am I audible? Hello?
Yes, sir. Yes, sir.
Yes, sir, you're audible, sir.
Yes, sir. Yes.
So the next
Audible.
part is planning and design of experiment. Everybody plans, everybody designs an experiment. But from where we get excellence? Let us see. See, whenever you are planning, what is there in your brain? Let's say I want you to synthesize aspirin. So what you will do? For the first time in your life, you are supposed to execute synthesis of aspirin. What you will do? First thing is mission statement. What is your mission statement? If you have a very clear mission statement, you are 100% sure that success will come. Mission statement is what you are supposed to do. And then what next we will do? Procurement strategy. In case of procurement, you need to be very sure that for your research work, ample material is available with you, because in R&D success rate is less than 30%. To be really frank, it is 18 to 20%. So that's the reason why don't think that whatever you are doing will always give you positive outcome. No, it is not like that. Out of 5 experiments, 4 fail. Okay, so that is what normally happens in life sciences, chemical sciences, and pharmaceutical sciences altogether. So that's the reason why you need to ensure that you have sufficient inputs to execute the reaction or formulation or biological activity. Let us imagine that you are supposed to screen anti-inflammatory activity. For that, you require, let us imagine 10 rats are needed. 10 rats are needed. Okay. So there are 100 factors are there where survival of a rat is there, responsiveness of a rat. So many things are there. So that's the reason why will you buy only 10 rats or 15 rats or 20 rats? It is better we have as many rats as possible so that we will have assurance that for our experiment, even if it is a failure, we will have another batch of rats are available for next try. Okay. Let's say you are synthesizing aspirin. You are asked to synthesize 1 gram of aspirin. How much salicylic acid you will procure? Will you procure 800 milligram exactly? No, it is not like that. You just buy 10 grams vial or 200 grams vial because it is very cheap. Okay, so you need to have plenty of starting inputs. So thereby you are confident that even if 1 or 2 failures are there, you can go ahead. Otherwise, what will happen? Something is actually dragging you back, thinking like, okay, if I use these animals, what will happen tomorrow? If I use this chemical, what will happen tomorrow? If it's a failure, what will happen tomorrow? It is all back that seriously affects your quality of research work. The next one is risk identification. Normally, what happens is in the laboratory environment, whenever you listen to any colleague, people will say like, okay, I'm putting this reaction or I'm doing this formulation, we'll definitely get it. But there is always a risk factor. Okay, we always have to see that risk assessment is done on every research work that we are going to pursue. Then project controls are there. So what are the points that we need to control so that we can execute the project properly? Then resource planning. Resource planning is again another one. Knowingly or unknowingly, visibly or invisibly, we have enormous resource around. Okay, so let us imagine that a chemistry person is synthesizing some scheme and they are supposed to do antibacterial activity. Okay, antibacterial activity, you need not send it to in your B.Pharmacy college. You have a microbiology laboratory where cultures are available, and there are experiments where in biochemistry, in microbiology, you are supposed to do antimicrobial activity. That is a regular experiment. That's a resource. Okay, so only thing that is needed is you need to plan so that your colleagues are informed, your inputs are provided to them, and you have proper terms of collaboration. So thereby you can do resource planning so that the flow of work is not affected. Flow of work is not affected. Then comes commissioning and validation plan. Whenever you are commissioning, it means initiating the research work, you always have to do the checks and balances all the time. Validate the protocols, whether everything is going on properly or not. Things need your attention. Okay, so let us say you are putting a reaction, you need to see that it is monitored properly. You are doing some animal experiment, animals, you have to see that animals are monitored properly, their environment, their food, everything is to be monitored. Your 100% energy you have to spend on the experiment that you are doing, then only high quality results you will get. Then the plan. Okay, once one experiment is done, then you have to plan for the remaining, so that one proof of concept kind of an experiment, once it is done, you have to extend it further so that the remaining experiments are done on the same line. So when you are doing it like that, uniformity is maintained. So as per your plan, everything will happen. Then you need to see the scope. Whenever you are executing an experiment, your scope of experiment will usually, you have to recalibrate your scope of experiment, because sometimes your expected outcome doesn't come. Something more, something less will come. So you have to see that at the end of the day, the scope of your experiment is recalibrated. And then design plan. Designing of or plan design you will do in the beginning, and then according to the recalibrated scope, you have to redesign your plan. Okay. Project execution approach is also to be properly monitored. Okay. So this is what exactly changes from person to person. If you really want to have high quality outcome, research excellence, you need to be focusing on every one of these aspects whenever or whatever the research work you are performing. The next one is execution of work. So how do you execute? While executing, first thing that you need to be very, very clear of is you will always have a research team. We don't usually work alone. Some of the things are there which we do alone, but there are things, many things where we need to communicate properly with our team members. We need to explain them what exactly is expected out of them, what exactly they are supposed to perform, so that they are very, very clear. Each one of them, starting from the group leader to the bench chemist to the sweeper, whoever is taking care of it, to the supporting staff, everyone need to understand what exactly they are supposed to do. That is what we call it as communication. You need to manage that properly. And then time management. Research work happens only when you perform it as per your time schedule. The moment you follow or you are maintaining your time, so then there is a possibility that you will have appropriate results in appropriate time. The next one is cost management. Finances or financial struggles are the major impediment as far as research work is concerned. Okay, so in the beginning itself, while during doing the planning, you need to have allocations for regular research work, as well as some of the money or funding you need to keep for emergency expenditure. Cost management is also very, very important. Then quality management. Quality management means, let's say when you are instructing something to the scientist, researcher, your student, or you yourself, you are performing an experiment. Okay, then how do you maintain your quality? For example, you are weighing some sample on a balance. Okay, then up to what digit you will record in your observation notebook? How do you maintain your observation notebook? All those matters. Okay, so quality is quality. So you need to be very, very sure about the quality parameters that you are defining to your team members. Next one is change management. Research work, as I told you all the time, has only 20% success rate. It is because it is always unexpected sometimes, because knowledge-based decisions we can take, but many a times they backfire, they fail. So change management means change in situation, change in protocol. When something goes wrong, what to do? You need to have plan B ready all the time. Next one is acceptance management. In case of acceptance, what do you accept? Do you accept a mediocre result? Do you accept result up to 20% error? Do you accept result which is of no consistency? Do you accept fudged data? All right, okay. So this is the way how. What is your acceptance criteria? You need to manage that. Procurement management, because whenever you are executing something, you need to procure certain things. Sometimes you need to procure chemicals, sometimes you need to procure manpower, expertise. Many of the things are there. So when you are in a position to take something from others, from whom you will take? From from which resource you will try to take something. Okay, so that is very, very important. Then issue management and risk management. Issue management means if something happens, how do you in response? So here also one need to have defined methods, okay, defined rules. So thereby, if you follow all these, then you can expect highly dependable, high quality data from your research team and also from your own research work. Then the research work, whatever you have done, is bound to have excellent outcome. Okay, so excellence comes when somebody is following all these criteria. And then data analysis and interpretation. Hello, am I audible? Hello? Okay. So in the data analysis and interpretation, once the experiment is designed, experiment is executed, now you have the ultimate role to play. That is, analyze the data and interpret it. So once an experiment is done, how do you understand the results? You got the data in terms of numbers. If you are synthesizing something, you get melting point, boiling point, theoretical yield, practical yield, percentage yield, or if you are doing dissolution apparatus, using dissolution experiment or some kind of BA/BE studies, biological equivalence studies, always you are presented with the data. So how do you handle that? So in case of data, we need to be very, very sure that integrity of data is maintained. So here, data collection and pre-processing. Data collection starts with you collect the data from absolutely from validated resource, and you will ensure that you are not considering irrelevant documents, including fudged data. Build term document matrix, weight term document matrix, because these are the technical terms that we use, so that whatever output that is coming from highly dependable source is always given high priority. Let's say some data is coming from an instrument which is validated, like HPLC, LCMS, or some other instrument which is validated. So those data are free from human errors, but you are getting data from anti-inflammatory activity of rat paw edema, because it is a human that is actually taking care of taking the data from a source. So human error is a possibility there. So you have to be very sure, cross-verify whether the data that is provided from non-reliable or low dependency level resources. Then singular value decomposition and analysis and interpretation. Here, these are technical terms that we use, where you need to see what exactly is the factor that you are supposed to focus on, so that the result that is obtained can have significant impact on your decision making. On your decision making means you started your research work with a certain presumption, certain plan, certain objective. So from the data that you obtain, which data actually suffices or helps you in solving the problem? You need to see that that value is, or the parameter is actually brought out. And then analysis. How do you do the analysis? In the analysis, one need to be really careful about using tools so that you will get meaningful outcome. Let us say single or one variable, one standard kind of a database is there. So you will be using one type of statistical operation to bring out correlation between a variable and a standard. Okay, so only two variables are there. When multi-data is to be analyzed, you need to use some other kind of statistical tool like partial least square analysis or something like that. And one need to be very, very sure if any other specific statistical tool is available which fits into your analytical requirement and obtaining high quality result. Okay, so one need to be very careful. It is not like somebody has done student t-test, somebody has done R-square value, somebody has done partial least square analysis, somebody has done some analysis, so I will also implement it. No, it is not like that. I often hear people saying, I am following a publication, I am following a publication. No, that is not it. You are supposed to define your own rules and regulations in picking up the data, in analysis, in choosing the right tool. Okay. If things are not clear to you, because ours is an application based science where research work quite often demands collaboration between various fields. So it is better you talk to an expert. Absolutely fine. The next one is revise and renew, how it contributes to excellence. Why do we have to revise? See, when you have executed something, you will have these outcomes like great outcome or poor outcome. It could be because of great process or poor process. Okay, so here when you observe 4 types of scenes you can see. The first one is you have implemented a great process and you got great outcome, because in your laboratory when you are doing your B Pharma, M Pharma, or when you are executing labs, you might have seen this. You have given your students great process. You have students who are dedicated, who have put in great efforts. So they got great outcome. Yeah, you got it. Second case is you will always find in your team certain people who does not follow your SOP. They don't bother about following SOP. They tweak, they do manipulation, but sometimes they get great outcome. Okay, it is only luck. So when somebody comes to you with a great result, stop and think. So the third outcome is you followed the great outcome, great process, but you did not get the result. Okay, so there is something seriously wrong with the inputs. And then you followed poor process, you ended up with poor outcome. So here comes again, you need to redo everything. Okay, so these are the 4 results that are expected in a research work. You have done an experiment by following a protocol. These are the 4 outcomes that you can get. So whatever the outcome may be, don't believe in the outcome straight away. When an outcome is there in your hand, and when the outcome is telling you something, sit and listen. Okay, so if you get a great outcome, whether the great outcome is because of great process or because of luck, that you need to assess, so that when a student or a team member or you yourself execute something and you got very good result, okay, so what exactly happened, you need to assess, because this is science. Sometimes if you depend on luck, in the next experiment it will be a failure. It will be a failure. And on the other side, let us say if something goes wrong, a researcher is there in your team, you yourself is genuine, but you have executed as per the protocol that is obtained from reliable resource. Quite often it happens when you blindly follow research articles. So here what happens is you followed an article that is published somewhere, believing in, okay, this is a very good article published by a renowned journal, and you have executed it, but you did not get the result. Okay, so quite often you will stumble upon these type of problems. we have faced such a n number of problems like this. Somebody says highly lipophilic molecule can be treated with some other compound and we got the product like that. But when we try to execute it, we are unable to get anything out of it. Okay, so we need to sit and do the assessment. We have to keep our beliefs aside and try to be assertive in executing a research problem. Then you will be able to understand the reaction, understand the success or failure, so that if needed, we need to revise the protocol, we need to renew our thought process. If you are open, if you have this capability, then only your research work can have excellence. And then comes excellence, publishing, patent, or making a product. Okay, so here again, if you really think like you wanted to claim yourself excellent or doing excellent research work, you need to be really careful as far as publishing, patenting, making a product is concerned. Normally, for an outsider, if something is of useful, we need to see these three things. One is cost, second one quality, third one speed. If you have done something, you have developed a synthetic reaction where you reduced the cost, increased the quality of outcome, reduced the speed, increased the speed. So that is what you call it as a value addition. Value addition. So when your publication or when your research work has this value addition, obviously people are there to recognize and appreciate it. Recognize and appreciate it. And also please see that other way. So we look for cheap processes, means affordable, good outcome, fast. So you have offered cheap outcome and that can be done quickly. You are compromising on the quality. If you have developed a cheap process and a good outcome, will take time to deliver because you are trying to reduce the cost so that it is made affordable to everyone, and a very good outcome is what you are aiming at. So it takes time, okay, so that you will have to wait. And a very good outcome very quickly you have to get, it will definitely be highly expensive because you are using all your resources in a short period of time. So they are they're supposed to be highly expensive. And when you are looking for an impossible situation where cheap, excellent product with which can be obtained very quickly, it is not going to happen in our R&D laboratories. Okay, so here these are the facts which one need to understand. Claiming something which they don't deserve, claiming something which did not happen, claiming something which can never happen. These are the three things which one need to avoid so that quality of your research work will improve immensely. If the quality of research work improved, people will appreciate it and your publication will have certain value. When you are publishing, what is your objective? That is what you have to define first. When you are publishing, your objective is to spread knowledge. When somebody keeps your research work in their journal, now that is open to the world. Okay, so some scientist somewhere, when he is looking for some issue which is nagging him, maybe he may find solution in your research paper. So that is the way how you are spreading knowledge throughout the world. Okay, when you have isolated a compound for the first time, please understand the value of that exercise. Okay, you have added something to the existing knowledge. You have synthesized something, you have formulated something which was never done by anybody else. So they are all of highest value. People will appreciate it. So be patient and see that your work quality is improved by avoiding false claims. So friends, these are the things that I just would like all of you to understand and see that you are implementing all of them in your day-to-day research work. So I found there are certain key ingredients are needed in a person to see that excellence comes out of their research work. So excellence is being ready to put in hard work and also to do smart work. Okay, hard work is needed sometimes, but doing the work in a smart manner, it adds a lot of value. This tendency is needed. We need to see that we are updating our capabilities. If a process is there where we can achieve something employing technology, smart methodologies, we can adopt them to our research environment by taking actions. Second one is perseverance. Okay, as I told you, in research we can hardly see more than 20% success rate. In case of synthetic organic chemistry, it is a well-known fact that the percentage is mere 15 to 16%, very, very low. To a failure, how do you respond to a failure? That's a very important trait. You really want to do excellent research work, you need to have patience, perseverance. So when something goes wrong, simply sit down and do the analysis where exactly what went wrong. So you find out the solution, but don't stop it. Patience. So this is the most important quality a researcher and every member of a research team need to have, so that excellent research outcome will come. Let's say if you have patience, you will wait for the animal to recover. You will wait for the reaction to happen. You will wait for appropriate chemical to enter into your lab. You will wait for a predetermined time period to complete. Okay, our gurus used to tell us, like, for a baby to be born, one need to wait for 9 months minimum. Okay, so if you don't have patience, I wanted a baby at 6th month, what will happen? You are destroying a successful process. Okay, that is true even for a research. Everything has its own time. In your planning and design of experiment, mostly by using your knowledge based decision, you will have that idea, this how much time you are supposed to wait, and you be open to the challenge. Sometimes it will not happen, give it some more time. Patience is a very important criteria, very important characteristic everyone need to possess. Next, courage. Friends, this is where I just would like to share with all of you that being a researcher itself is like living in hell. Okay, because you are under pressure. Why are you performing research work? Your objective, let us say, if it is to get a publication. So if you are publishing, what for you are publishing? For your career development, for building your portfolio, for building your CV, or maybe your college management needs high journals. So obviously you are under pressure. When you are under pressure, what you will do? You have to do something so that their needs are satisfied. Okay, but have courage to convince yourself, convince your team members, convince your colleagues, convince your management. Have courage to see that your plan is executed properly. Have courage to see that you are fighting against odds. Have courage to see that you are abstaining from abstaining from spoiling the environment. You are abstaining from malpractices, abstaining from shortcuts. Have courage. Okay. So this is again very thing one need to possess so that you will have excellence. Integrity. This is again trait which I have seen wherever and whenever excellence is there. In that research team as such, research members, team leaders, everybody has integrity. So I can tell you 100 different examples where because of lack of integrity, failures happened. In research work, okay, let us say some of you might have communicated papers with various publishers. Then when you are communicating a research paper, let us imagine you have done some method development using HPLC. You have incorporated tables containing data, figures with chromatograms. Do you think so the editor will verify everything? No, it is not like that. Editors will usually have faith on researchers and faculty, academicians, because the unwritten rule here is they believe in we performing research work and producing results. Okay, so here that integrity component is what exactly is making the situation happening in a smooth manner. Let us imagine that we lose our integrity. What will happen? You communicate a paper to a publisher like a Springer or American Chemical Society or somebody else. Then for a HPLC method development, then the editor will ask you, will send you a letter saying, sir or ma'am, please send me your sample and also send me $1,000 so that I will send these samples to local pharmaceutical analytical laboratory. We can cross-verify your data. Cross-verify your data. The moment we lose integrity, this is what is going to happen. So if you don't believe my words, I'll give you one example. We have GVK Bio, that is a pharma company. Okay. Do we have GVK Bio right now in the bioanalytical division? No, that is closed in 2010 or 2012. Why it is closed? Because they were fudging the data. They have clinical data with them done by some of the analytical chemists who cannot afford to say no to the pressure. Their higher ups are saying like the values with you are 98.9, but what we require is 99.1. Only 0.3 difference is there. You fudge the data. That is the pressure. That is the kind of shortcuts that are offered. And they kept on implementing this fudging of data, which was later found by US FDA. And then they slapped the company with a fine of 500 crores. With that, they had to close the company. Now what US FDA is doing? The moment an Indian company send papers for them for approval, they are cross-verifying everything with the local labs. Okay. The moment you lose integrity, all your actions are questioned. Okay. So that is where we need to be really, really careful. I am afraid soon this kind of a situation will arise. We need to strive very, very hard to see that that kind of situation does not arise. The next one is attitude. Okay. Excellence comes with the personality. Every person, it is like more like personal thing. Like when a researcher is entering into some kind of a task, what should be his or her attitude towards execution? They need to see that 100% effort is kept in executing a particular task as per SOP, and you shall not in whatever the circumstances may be, you shall not deviate from the process. You shall not deviate from the process, especially in the research work. If you cannot implement your protocol, please don't execute it. Okay, so that should be the attitude one need to develop, so that you yourself can be satisfied with the outcome, and also you are adding a lot of value to the scientific world. Okay, sometimes what happens is you made a compromise in a specific data. The data is now published. Somebody tries to implement it in their laboratory, research lab or production lab or whatever, and they were unable to implement it in whatever way that they try. What will happen? That's what we call it as polluting the literature. Okay, so we shall avoid such a thing. So this comes from attitude of an individual, attitude of the research team. So friends, these are the key ingredients one need to inculcate to see excellence in their research work. I will conclude my session with a quote. Excellence is the gradual result of always striving to do better. Okay, so if we are able to implement this, we can achieve whatever that we are planning for. So friends, I thank you all for your patient listening. Now the session is open for discussion. Hello.
Hello. Thank you, sir. Thank you for an
Yeah,
informative session. If anyone having doubts, you can ask the sir.
Sir, in a pharmacy field, hello, can you hear me, sir?
Hello. Yes, sir, I can hear
Sir,
you.
sir, what are the software you can use for the as a researcher in pharmacy practice for doing good research? What are the softwares?
One second. The selection of a tool depends
Yes.
upon the topic that we have chosen.
Okay. You
So
have
in
any
pharmacy
idea?
practice is a ocean. Okay, so let us say you are looking for or pursuing a problem where you are looking for data of patients or a database containing various kinds of information. Then you need to look for appropriate database that is available or not. For PharmaD, we have a couple of databases.
Okay.
So if you can use it, then you have to see that. Second one is, in case of pharmacy practice, let us say you are into clinical segment. In case of clinical segment, whatever are the top publishers that are there in the world, you need to see, because it all depends upon the kind of topic that you have chosen. You need to see, because we are fortunate that internet is there. You need to see that those publishers' website is approached and search there for relevant data. It is not like always a software or a database is available. No, it is impossible. It is because PharmaD course is there. That's the reason why we have certain databases that are available right now. Otherwise, we would have to depend upon online resources only.
Okay. Okay. Thank you, sir.
Okay, thank you. Any others? So I have a word of caution for all of you. Hello.
Yes, sir. Yes, sir. Tell me, sir.
Okay. So in the recent times, it so happened that the reason why I accepted this talk is like research excellence is one of the word that I mandatory that we have to go for NAC accreditation or NBA accreditation or one or the other rankings. So that is what made many people to realize that publications are needed. But the problem is research cannot happen overnight. A faculty who stays in the college or stays with the college for at least 4 or 5 years can build a platform on which he can grow. He or she is supposed to work and build a research team, research method, research environment, and then that environment will give you compounding effect. In the beginning, if you start the research work, maybe you may take 1 year, 2 year, 3 years to produce a paper. Don't worry. And after that, every year you can publish 2-3 papers because the platform is already ready and students are there. And I can see clearly there is immense pressure from college managements because it became something like we cannot go back. So they are pressurizing the faculty to produce high impact, to quote, high impact journals. For publishing in high impact journals, let's say if you have proper research facility, it will definitely take certain time. In biological research work, it may take 2-3 years to give out a high impact journal. In computational field, at least 1 month or 2 months are needed, dedicated time, so that you can produce something. But per month 2 publications, in 3 months 5 publications, this type of calculations there doesn't work. Okay, so I request all of you to please see that these are all practicals. When somebody asks you to publish, please see that, have courage to say it is not possible. Okay, convince them, no problem. Second thing that I have seen is, maybe some of you are having WhatsApp groups where you are forcibly added by some of the WhatsApp groups like publication group, book publication group, patent publication group, where I have seen such a pathetic situation where the authorship is sold. Authorship is sold. Like you don't have to do any research work. You only have to buy author position. So where are we going? Where is the excellence? Let us say you have bought another publication in PLOS, Class A, that has impact factor 6. You try to buy authorship in Class A. Okay, so that fellow will ask you, what is your contribution? They will verify. Okay, they'll ask you a contribution. Okay, so you are in a position to be asked by somebody else. Second thing is, if you are entering into such a kind of a deal, you are forcing yourself into a situation where you cannot recover, because somebody gave you an offer. So you bought a other position in statistics journal, chemistry journal, analysis journal, biology journal. First thing is, do we have to do that? Is it ethical? Second one is, patents also is like that, and book chapter is also like that. Somebody is selling. So let us say if you claim that I have Class A paper is there with impact factor 6, do you think morally it is correct? Excellence doesn't come in that way. Maybe your numbers are correct, but if you have that kind of attitude, every one of us have to see that that practice is now, we have to nip it in the bud. Okay, so there is no other way. It is rampant, and people are in a hurry to fill their CV with high impact journals by simply selling their property and giving all the money to the publishers. This is a kind of regressive thought process. Okay, please see that, please take it in the right perspective. I have seen that. Please see that you are not encouraging this kind of environment, because when you utter the word excellence, excellence will come out of right kind of environment. I thank you for accepting my request and letting me speak a few words.
Thank you, sir. Thank you so much for your message. Now I would like to extend my sincere gratitude to our esteemed guest speaker, whose presence has added immense value and prestige to this program. Your insightful remarks have inspired us to strive for the excellence and have provided us with invaluable guidance for the journey ahead. On behalf of CREDDY College of Pharmaceutical Sciences, I extend my heartfelt appreciation to all the individuals who have made this event a resounding success. Thank you, everyone.
Madam, can I leave?
Yes, sir.
Hello,
Yes,
Lavani.
Sir, Murli, sir, good presentation, sir.